Good morning, all. Uh, I hope your day is starting well. Uh, is there anything anybody would like to talk about before I start the lecture and with the reminders and everything? Okay, let's get started. So, the usual reminders. We have quiz 4 that is open, and you are welcome to take it anytime between today and Monday, February 22nd. It is due on Monday. Again, we have a little break from our usual quiz routine, but we will go back to the usual next week. So quiz 5 opens on February 23rd, or technically on at midnight on February 23rd. So after quiz 4 is due, quiz 5 opens, and you can take it anytime from February 23rd until Friday, 11 p.m. on February 26th. There is a participation assignment that is due today. There are 3 questions. They are drop down, true, false, and multiple choice question. Should be fairly simple after the lecture. Yeah, so don't forget to take it. It is due today before 11:59 p.m. The topic of today's lecture is going to be the nervous system. We are venturing into studying human organ systems, and not only human, but really animal organ systems, because nervous system is fairly similar between animals. Well, mammals, I should say. Um, and nervous system is fairly complex. It consists of around 100 billion neurons. And again, neurons are cells that nervous system is made up of. So neurons simultaneously control whether we are awake or asleep. They control our muscles. So whenever we move our muscles, that's because the neurons kind of whipped the muscle and caused it to move. Um, so, despite the complexity of the nervous system, all neurons work in a similar way. They send electrochemical signals from cell to cell, triggering instantaneous responses or instant responses. So, today I will cover basic structure and functions of the nervous system and senses, such as sight, hearing, smelling, and sense of touch. Uh, what was surprising personally for me to find out, I always thought, well, not always, but up to a certain point, I thought about nerve impulses as being electrical impulses. So I thought they were traveling at the speed of light. And it is not true. They travel much slower than the speed of light. It is still pretty fast, but not as fast as the speed of light. And in fact, I will go over today. Today, I will go over the methods that would allow you to actually measure how fast do your nerve impulses travel within your body. So the vertebrate nervous system is fairly complex, and this picture kind of shows what is going on, how animals process the visual and auditory information and olfactory information and react. So animals such as lynx and rabbits shown here rely on their nervous system, rely on their abilities to detect stimuli, visual, auditory, or olfactory, and coordinate responses, form memories, solve problems, and communicate. So, despite that, the picture may be pretty boring. What is going on in the lynx's nervous system or in the lynx's body at the time is all but boring, and it's fairly sophisticated. So the lynx kind of when the lynx sees the rabbits, the lynx processes the visual information, the auditory information, and the smells of a rabbit, olfactory information. So the information is perceived by eyes, by ears, and by nose. The impulses travel towards the brain. The brain processes the information. So the lynx decides, ooh, food, I better go after it because I'm hungry. And In the brain, once the response is formed, the brain sends the impulse to the muscles, allowing the lynx to pounce at the rabbit. Okay, so the general processing information processing pathway is from the senses. The information comes to the senses, to eyes, to ears, to taste receptors. Maybe if you're eating something, it goes to the brain. The brain processes the information and formulates a response. You may do nothing, or the brain may decide, okay, since you're hungry, you may go after that hare. And the response is sent via neurons, and maybe it will be muscles that will be engaged, maybe it will be hormones that will be released, and so on. Often it's both. The nervous system forms a rapid communication network. So there is rapid communication between cells, and it is fundamental to the function of the animal nervous system. We absolutely need to react fast, otherwise we won't survive. Well, it was in the past, but in the past, if you didn't react fast, you wouldn't survive. And even now, like if you touch something hot, you need to stop touching this hot thing that is damaging your tissues, and you need to do it fast. So nervous system kind of is adapted, it has evolved to facilitate this such fast reactions to external stimuli. So in the picture here, you see a neuron, and the neuron, I will go over the neuron structure in the next slide, but besides the neuron, what you can see here is myelin sheath. And this myelin sheath, the function of this myelin sheath is actually to speed up the travel time of the neurochemical impulses from one neuron to another neuron. If there was no myelin sheath, then the reaction time would be much slower. In fact, some of the diseases that result in the degradation of the myelin sheath result in exactly that: slowed reactions, slowed responses, and slowed cognitive ability of people whose myelin sheath is damaged. And myelin sheath itself, if you're wondering what it consists of, it consists of water, lipids, and proteins. Yeah. Now, neuron itself consists of three parts. There is cell body that has a nucleus. It has dendrites, or the part of the neuron that receives electrochemical impulses. And it has a longer part, a longer tail here, called axon. So the longest part of the neuron is called axon, and this axon is covered by the myelin sheath. Now the axon can communicate the information to or pass the electrochemical impulse either to a muscle, as this picture shows, or to another neuron, or to a gland. And I think that's about it. So a neuron can pass the information to another neuron, to a muscle, or to a gland. If the information is passed to the muscle, if the electrochemical impulse is passed to the muscle, then the muscle starts working, starts contracting and releasing. If the information is passed, if the neurochemical impulse is passed to the gland, the hormone start being produced. And if the information is passed to another axon, it just travels somewhere in the body where it's needed. All right, I see some chat questions. Ansley's got it. Thank you, Ansley. Okay, so 3 parts of the neuron. Neuron consists of 3 parts: dendrites, which is the receiving part of the electrochemical impulses, the cell body, where the nucleus is located, and the axon, which over which the information travels either to the next neuron, the muscle, or the gland. Okay. Now, to the interesting part. How do these electrochemical impulses are generated in the neurons? And if I summarize it on a high level, these electrical impulses are generated by using the proteins embedded within a membrane, potassium and sodium ions, positively charged particles, and negatively charged proteins. Okay, so neurons work to keep potassium concentration higher inside the cell than outside, and sodium concentration higher on the outside cell than the inside. So in this picture, you will see the inside of the neuron, or specifically inside of the axon, outside area from the neuron, and this inside and outside are separated by the membrane. By the membrane of the neuron. So from previous lectures, we know that there are proteins embedded in the membrane that have various functions. And this particular protein should look familiar because this is the sodium-potassium pump. So sodium-potassium pump assists in electrical impulses that are generated within a neuron. So inside of the axon, or inside of the neuron, we see more potassium compared to the outside of the neuron. And there is more potassium on the inside, partially because of the sodium potassium pump, but also, sodium potassium pump actually is responsible for the more potassium on the inside of the cell, because sodium potassium pump pumps the potassium inside the cell and expels the sodium to the outside of the cell. Now, another part that we did not see before are these negatively charged proteins. It's not quite important what they are for the purpose of this class, but it's important to know that they're there because these negatively charged proteins, they facilitate the negative charge close to the membrane on the inside of the cell. So this pipe here is the schematic representation of the axon. So on the inside of the axon, we have a negative charge, and on the outside of the axon, immediately on the outside, we have a positive charge. And as a result, we have some voltage in the axon. So if you had a voltage meter, you would the inside of the cell would be negative. Right, I have some. Lauren is asking, this is an active transport in the sodium potassium pump? Yes, this is an active transport. So ATP, which is pictured here, is needed to make the sodium potassium pump work, to pump out sodium and pump potassium inside the cell. Ryan asks, so are myelin sheath considered catalysts? Not quite. Catalyst is used in the chemical reaction, okay? So it's not, like enzymes are catalysts, but myelin sheath is just a structure that facilitates faster conduction of the electrical signal. So it's not quite a catalyst because it's not a chemical reaction that is going on. And Ansley just repeated that. Thank you very much, Ansley. Okay, what else do we have embedded in the membrane? We have potassium leakage channel, which is always open. And the potassium will move either inside of the cell or outside of the cell, depending on where the concentration of potassium is larger. In this particular figure, the potassium concentration is higher on the inside of the cell. So the potassium will move out of the cell or move down the gradient of concentration. If the concentration of potassium becomes higher on the outside of the cell, the potassium will go on the inside of the cell down the gradient of concentration. And we have two other proteins embedded here, one of which is the sodium channel. It remains closed unless something stimulates the neuron. And we have delayed potassium channel, which is closed until something stimulates the neuron. Okay, so those are the things that are embedded within the cell that take active part in the generation of this electrical impulse. So what's important to remember is that the inside of the axon cell is negatively charged, the outside of the is positively charged at rest. Okay, and why is it important to remember is because in the next slide I will show that what is this electrical signal, how it is generated, is by change in the voltage within the cell. And this change in the voltage within the cell is produced by opening of the sodium channels. So whenever a neuron is stimulated, the sodium channels embedded within the axon, the neuron membrane, they open and let the positively charged sodium inside of the cell. So in the purple here, we see the increased sodium concentration within the cell. Because sodium is positively charged, the charge inside the neuron becomes positive. And here's our old voltage meter here, and it moves closer to zero. So the more stimulation is applied to the cell, the more channels, the more proteins embedded, the more sodium channels embedded within the cell membrane open. And they open for a longer time if the stimulation is continuous. And sodium ions enter the cell and collect near the membrane, reversing the charge. So it's basically what is depicted in the schematic axon here. So the charge inside the neuron is switching from negative to positive. And this is the beginning of our electrical impulse, okay? This change in charge, it is kind of information that will get passed on to the next neuron, or to the muscle, or to the gland. So sodium ions accumulate to start an action potential. Once the action potential begins, more sodium channels open, allowing more sodium ions into the cell. So the membrane now has a positive charge on the inside. So action potential result from changes in the electrical charge. You will probably see that on quiz or on the exam sometime down the road. So the action potential, the definition of it, is the change in the electrical charge inside the neuron. So in the previous picture, in the previous slide, we see that inside the axon, we have a positive charge on in this location, but in this slide, we see that this charge somehow moves along. What is going on here? This charge somehow moves along the axon towards the next cell. So how is this movement happening? Because if the nerve is stimulated, why don't all of the sodium channels open at the same time? Or well, they are kind of they do, but the electrical charge, the negative electrical charge, is immediately reestablished inside different parts of the cell. And this is what allows this immediate reestablishment of the negative electrical charge is what allows the action potential or this electrical impulse to travel down the axon and then on to the next neuron or to the muscle or to the gland. So what reestablishes this negative potential is these delayed potassium channels. So potassium, which is ions of potassium, which are positively charged, diffuse out of the cell as sodium channels close, returning the membrane to its resting potential. So first, sodium channels open, and there is a positive charge. And when the sodium channels open, potassium channels open. Okay, so one positive particle is flowing in, positively charged sodium is flowing into the cell, but at the same time, positively charged potassium flows out of the cell. So and this difference in the flows of the two positively charged particles kind of even out eventually, and the charge on the inside of the membrane returns to its old state, to being negative. So despite that no negative particles are flowing in or flowing out of the cell, the charge can be reestablished by expelling more positively charged potassium particle atoms out of the cell. And eventually the sodium potassium pump will get rid of all the excess sodium inside of the cell. And that this is how sodium gets eventually out. This is how sodium that entered through the sodium channel gets out of the cell.
Professor, there's a question about the receiving cell and how it's affected by that sodium influx. Is there anything important?
We are not there yet. So
Okay.
the way that it travels to another neuron, we're not there yet. I will get there. But
Joshua just pulled.
At this point, we're looking just at how electrical impulse travels along the axon. Okay, how it actually may be received from the dendrites and how it travels to the axon. Now I'm looking through the chat here. The question is the receiver. Oh, okay, that's the old one. Now neurons pass the message or this stimulation along either to the muscle gland or next neuron. And I'm going to talk about first. I'm going to talk about how these electrical impulses are passed towards the next neuron. So neurons form a communication network by passing information to other cells, and this is accomplished in two ways: by electrical impulses, this is first way, and chemical signals, this is a kind of. So neurons pass the message from cell to cell, but they don't technically touch each other. Neurons do not directly connect to each other, okay? They use neurotransmitters, which are chemicals, to pass the signals. So examples of these neurotransmitters are histamine, serotonin, gamma aminobutyric acid called GABA. So histamine, for instance, you're probably very familiar with it if you've ever been allergic to something. The allergic reaction is triggered by release of histamines, and this release of histamines causing increased heart rate, swelling, dizziness, and so on. You probably have heard about serotonin before because of the popularity of antidepressants nowadays. So serotonin is so-called a happiness hormone, and it's kind of calms down the nervous system and makes you feel happier. And gamma aminobutyric acid, GABA, is the kind of inhibitory neurotransmitter that I'll mention it again a bit later. It promotes sleep and it helps us relax. GABA is kind of the brakes of the nervous system. So neurons do not directly touch other cells. Instead, they form a structure called a synapse. And a synapse is basically this place in the neurons where they come together, quote unquote, but not directly touch. So in the synapses, the chemical signals or neurotransmitters are released. So I wonder, yep, the next slide shows exactly what's going on. So this this electrical impulse, this is the axon of the giving neuron, and this is the dendrite of the receiving neuron. So the electrical impulse travels along the axon and arrives at the end of the axon. And what happens next is, as a result of this electrical impulse, the vesicles containing the neurotransmitters, they are released into the space in between one neuron and another neuron. Okay, and once the neurotransmitters are released in this synaptic cleft or the space in between the two neurons, the ion channels in the next neuron, in the dendrites, they pick up these neurotransmitters and interpret it as a stimulation. So the sodium channels in the receiving neuron, they sense the neurotransmitter inside the synaptic cleft and open. And because the sodium channels open, the new electric impulse is created, which travels from the dendrites to the end of the axon of the receiving neuron, and this reaction, this impulse is propagated from neuron to neuron to neuron to neuron. So electric impulse does not travel directly from neuron to neuron. To sum up, it travels to the end of the given neuron, triggering release of the neurotransmitters into the space in between the two neurons called synaptic cleft. the sodium channels of the receiving neuron sense the presence of the neurotransmitter in the synaptic cleft, open, and the new electric impulse is generated. Is it more or less clear? Shout out a question. If not, Joshua asks, is the receiving cell affected by the sodium? Yes, the receiving cell is affected by the sodium. The sodium channels in the receiving cell open because they have sensed the presence of the neurotransmitter in the synaptic cleft. So Lauren says, I'm still a little lost. Do you mind go over it? Do you mind going over it one more time, please? Yes, tell me where to start, Lauren, from which part I lost you.
I understand like all the way up how like the sending neuron comes through, but really like looking at the diagram when it gets to like step 3 is where I get a little lost.
Okay, so step 3 depicts just the presence of the neurotransmitter, of a neurotransmitter inside the synaptic cleft, the space in between the neurons. And step 4 indicates that the sodium channel sensed the presence of the neurotransmitter in the space in between the neurons and opened.
Okay, thank you.
Is that better?
Yes, that's better. Thank you.
Okay, and any other questions?
What was the charge inside the cell at resting potential?
Are we talking about this cell or this cell?
The axon.
So the charge at rest, the charge inside the cell is negative, and the outside of the cell it's positive. But the electric impulse itself, it's like the traveling of the positive charge along the cell. Okay. So wherever the action potential is located, this is going to be the area within the cell with a positive charge, with other areas with a negative charge. Christine, did I did I answer your question?
Yeah, so wherever the action potential is, that's where it's going to be positive.
Yeah, it's going to be a region with a positive charge surrounded by the negative charge. Okay, Chantel asks, as the action potential moves from one neuron to another, does the amount of action potential change? It depends on the individual cells. I mean, I think so, because each cell is unique. One cell may have like more functional sodium channels, one might have less. So I suspect it might change, but not significantly unless there is some sort of something's wrong with the cells. Okay, so at this point, I wanted to talk more about neurotransmitters a bit. So what kind of neurotransmitters can be released into the synaptic cleft? There are basically two types of neurotransmitters. The first type is called excitatory. So they excite you, they excite the neurons. And the second type is called inhibitory neurotransmitters. And as the name suggests, they inhibit the activity of the next neuron. So the example of excitatory neurotransmitters is histamine and dopamine. The histamine, as I mentioned before, it's a neurotransmitter that triggers fight. I wouldn't say fight. It's basically makes us alert and triggers allergic reaction, which is an overreaction of an immune system. And dopamine is a pleasure neurotransmitter. It plays a role in how we feel pleasure. It affects learning. It affects motivation and movement. And because it affects movements, there's a disease called Parkinson's disease. And this disease is for this disease, it is characteristic for people to have low dopamine levels. And because they have low dopamine levels, they experience the symptoms of the Parkinson's disease, such as slowed movement, like tremors, and this uncontrollable movement of the muscles. So the examples of the second type of neurotransmitters, or inhibitory neurotransmitters, they basically, their function is to decrease the chance of action potential, and they are serotonin and GABA, or gamma aminobutyric acid. You don't have to know this. You don't have to know it. It's just you will see it, I think, more frequently as life goes on, and it's useful to know what it is. Like right now, there's these supplements in pharmacies called GABA, and what is it? Well, it's a neurotransmitter, but there's a whole separate debate on whether this ingesting GABA or eating it could increase the neurotransmitters in your nervous system, this particular neurotransmitter in your nervous system. And the research says that no, it cannot. If you eat GABA, it just doesn't cross the blood-brain barrier, and you just poop it out or pee it out. Nothing happens. No benefits are reaped from that. And the second one is the serotonin. Serotonin is considered inhibitory neurotransmitter, or the one that decreases the chance of the action potential. And interestingly, so I have this note on my slides here. The anticholinergic drugs that is basically antihistamines. Okay, they are allergy meds, heartburn meds. A study that was done about 1.5 years or 2 years ago found that increased dosages of the allergy medication and heartburn medication that suppress the acid production in the stomach, they can mimic Alzheimer's symptoms in the old people. So, and it happens because older people, our ability to metabolize these drugs, it decreases over time. So old people don't metabolize these drugs as well, and these drugs accumulate in the system. And because they suppress histamine and other excitatory neurotransmitters, older people may start experiencing Alzheimer's symptoms. And it was quite a discovery that led to revision of the dosages of these medications for the older people to accommodate the decreased rate of elimination. All right, I'm going to look at the chat for a minute, and if you have a question. Oh, Christine asks, would an EpiPen be considered an inhibitor? No. So EpiPen stands for epinephrine pen, and epinephrine does the opposite. So the way EpiPen works is kind of counterintuitive, but it counteracts the allergic reaction. It doesn't really calm you down because epinephrine is a stimulant, but it counteracts it helps calm down the overreaction of the immune system. All right, any other questions? Okay, so a little instruction for you, if you're interested in this sort of thing, is the exercise on determining how fast are nerve impulses in your own nervous system. So for this exercise, you will need a ruler, and I think like 30 centimeters will be enough. If you've got longer, great. But the way you will do it, you will need a buddy to do it. You can hold the ruler at the 0 mark, or rather your body, your buddy, your friend can hold the ruler at the 0 mark and let it out, and you would have to catch the ruler somewhere lower down the line, and you make a record of the length of the distance that the ruler fell towards the ground, and you would then calculate the reaction time or the speed that it took for your eyes to see that the ruler was falling, and for that information to travel to the brain, and from brain reacting and sending the information to your hand to grab the ruler. Okay, and it works better in person than online, but I think it would be something to do maybe on a Friday night if you're over 21 and have a beer. It could show you like by what percentage your reactions slow down after you've had a drink, or by what percentage your reaction went up if you had a good night's sleep, for instance. So the way to calculate your reaction time is through some knowns, which is the gravitational acceleration. When something falls to the ground, it accelerates with a constant speed of 980 centimeters per second squared. Now, you will have the distance that the ruler fell, or this distance between your body letting go of the ruler and you catching it, okay? And if you record this distance, you can then plug it into the formula and get your reaction time. And then watch how reaction time changes under different circumstances. If this is not clear enough, please drop me a message and I will draw a diagram for you or record a video how it's done. But it's important to have somebody else to perform the experiment with you, because if you perform it on your own, you will be kind of primed. It will not be a pure experiment, because your nervous system will be responsible for letting go of the ruler, and your own nervous system will be responsible for catching it, you know. So it's not going to be a pure experiment. You need somebody else to drop the ruler for you. So nervous system has two components: central nervous system and peripheral nervous system. And nerves are, the peripheral nervous system consists of mainly of nerves. And what nerves are, they are bundles of axons encased in connective tissue. In most nerves, the sensory and motor nerve fibers are bundled together into a single cable. So we have two types of nerves. The sensory nerves bring sensory information to the brain, and the motor nerves, which carry instructions for muscles and glands. So in our sensory neurons, they carry information towards the brain, and motor neurons, they carry information out of the brain to the muscles and glands. So let's say if you see something, your sensory neurons are engaged, and these sensory neurons bring the information about what you see to the brain. The brain then interprets the information, and if the brain decides to react in some way, let's say you see a bear, your sensory neurons are engaged and bring this information to the brain. The brain processes the information. Okay, I just saw a bear, I better run away. And this information, this decision that was made by the brain, is then passed onto the motor neurons. And these motor neurons, they can make your muscles work for you to actually run away, and they can increase the heart rate. So the reactions that you do not control, right? So the speeding up of the heart rate, release of the adrenaline that helps you kind of fight, that is responsible for fight or flight response. It is also stimulated by the motor neurons. So basically, if the nerves go to the brain, it's sensory neuron. If they go outside of the brain to the glands and muscles, it's the motor neurons. Now, the central nervous system consists of the brain and spinal cord. So there are two types of nervous tissue that make up the central nervous system: gray matter and white matter. Gray matter is the neuron cell bodies and dendrites, along with their synapses, or the connections between this connective area between two neurons. And white matter is the myelinated axons that transmit information throughout the central nervous system. And this is how the brain looks if you cut it in half. Uh, yeah, this is the look from the top. If you look at the head from the top, this is what the brain looks like. And if you take a cross section of the spinal cord, this is what it looks like. So this is our gray matter, and this is our white matter. And again, the gray matter is basically cell bodies with dendrites. And this is, by the way, where information processing occurs. And white matter is the axons with their myelin sheath. And in the brain, it's the same thing. There's gray matter and white matter. So white matter is, well, white, and gray matter is depicted as darker color. The brain consists of three parts. There's forebrain, midbrain, and hindbrain. So the hindbrain is illustrated in green. The midbrain is illustrated in this pinkish color, and the forebrain is illustrated in yellow here. And it's the name is literally because of the orientation, like the spatial orientation that the structure is located at. So in the hindbrain, we have a very important part. Well, all parts are important, but this part called the medulla oblongata is controlling our heartbeats, blood pressure, and breathing. So it's controlling some background functions of the body that are quite important and that you cannot really control consciously. Now what do we have? The pons, which is located right here, this little bit here. Its function is basically to connect forebrain with the medulla and cerebellum, or with the forebrain, basically. And the cerebellum controls posture and balance and coordinates subconscious muscular movements. So this part here. So as you may deduce, if it matters where you hit your head and where the damage occurs, right? If you hit your head really hard and you shake up or damage your cerebellum, you'll have problems with posture and balance, and you may have some problem with coordinated movements as a result. Now, the midbrain, its main function is to relay information about voluntary movements from forebrain to the spinal cord, which is illustrated right here, but it goes all the way up here. It's just hidden behind the hindbrain in the picture. Now, the forebrain consists of three major parts, and the first part is thalamus, and it is right this part here. The thalamus processes the information and relays it to the cerebrum. And hypothalamus, which is literally translated as below thalamus, it's responsible for homeostatic control of most organs. So hypothalamus, it controls the activity of our thyroid gland. Hypothalamus also, remember from the previous lecture, hypothalamus was controlled the temperature responses and so on. And finally, cerebrum over here, it consists of gray matter and white matter, and it's basically processes all other information. So creative thinking is done in the cerebrum, like creativity is in the cerebrum, and various other like stress response. Well, stress response all occurs in the forebrain, but a lot of the stress response or interpretations are occurring in the cerebrum. And it's 10:15. Are there any questions so far?
Can you just go back over the midbrain and like what that function is one more time?
Yeah, the function of the midbrain is to communicate the information about voluntary movements from forebrain to from spinal cord. So communicating the information from the yellow part to the spinal cord. And spinal cord is basically a bunch of nerves that will communicate the information to the motor neurons. So the spinal cord here, it's part of the central nervous system, but it communicates the information, it relays the information, it's connected to the peripheral nervous system. So if we look at this picture, the spinal cord is in the center, okay, and it goes from the brain all the way to here. And it is connected to the motor neurons and sensory neurons. So the sensory neurons, they bring information to the spinal cord, and this information then travels up to the brain, where it's interpreted and the response is formulated. Now this response travels to the spinal cord and then to the motor neurons. Was that helpful, Sarah?
Yes, thank you so much.
Okay. Any other questions? Comments? Hello? 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 Okay. Now it's 3 minutes left, so I'm not going to go over all of the senses, but I will start. So the senses connects the nervous system to the outside world. We sense the world through vision, smell, taste, hearing, and touch, temperature, and pain. Okay, the senses are very important parts of the nervous system. They include general senses that can be sensed all over the body, such as touch, temperature, and pain, and special senses that are restricted to the head. And these senses include hearing and equilibrium, vision, smell, and taste. So sense organs have sensory receptor cells that respond to stimuli. So sensory receptor cells contain a variety of different sensory receptor proteins. Well, you can read all that, but the gist is in the eyes, cells express photoreceptors, and there are two types of photoreceptors. There are so-called cones and rods. And I will go over what cones and rods do when I go over sight. But in the nose, for instance, there are different types of, this is ear, but in the nose, there's a different type of receptors that are called olfactory receptors. And basically all those receptors consist of proteins that respond to odorants. The olfactory receptors, that is, are proteins that respond to odorants. And the photoreceptors are proteins that respond to light. So you get the importance of proteins, or the gist of it. Okay, I'm going to go over mechanoreceptors. So the sense of touch comes from mechanoreceptors. The sense of temperature comes from the thermoreceptors, and we have a lot of those in our skin. Okay, the general senses detect touch, temperature, and pain. Each of these senses uses its own type of receptors. And basically, receptors are embedded in our skin and are often connected to the hair shaft. So whenever something moves the hair on our arm, for instance, we feel it through the hair follicle receptors. Now we have deep pressure receptors that are not connected to the hair follicles, but that are embedded within the skin. So if we press on the skin, we feel this pressure thanks to the deep pressure receptors. So the sense of deep pressure, or other like sense of heat or touch, or light touch receptors, they all are passed to the sensory nerve fibers and away to the brain. And then the brain interprets the information and passes this information to the spinal cord and to other muscles or glands in order to respond in some way. All right, it's 10:20, so I'm going to pick up the general senses and other senses that I did not cover during the next lecture. Are there any last minute questions? Christine, could you hang back? I'm gonna chat you back here.